Good morning, students. Last time we had discussed this DNA replication. Today we are going to say this new topic that DNA replication is semi-conservative. What does this mean? That if कंजर्वेटिव यहाँ पर अगर हम कहेंगे जो टर्म मीन करती है इसकी कंजर्वेटिव को, दैट मीन्स द ओल्डर. When it is the conservative, then we can say it is modern. Yeah, then we can say it is new rather. Now, when the term comes this semi-conservative, then it comes that it is half new, half old. There is the chances of 50-50. Chances nahi, 50% it is the new and 50% it is old. So, what we will study that this DNA replication is semi-conservative. That means that this DNA replication जो हो जाती है, it is what we call as semi-conservative. That it is half of it is what we call as this older जो इसका एक strand है, that it may be the new strand, and other strand जो है that it may be the new strand. Now there are some evidences. हमारे पास कुछ experimental proof भी हैं. They have got the evidences that this DNA replication is semi-conservative, and one experiment जो हमारे पास है that was carried out by Messelson, MESSEL, Messelson, और दूसरा जो इसके साथ है, his name was Stahl. So the experiment which was carried out by Messelson and Stahl, and they have done this experiment on the bacteria that is E. coli. On the bacteria E. coli. What they have done, this by carried out the experiment on this bacteria, on these cells of this E. coli, they have proved that DNA जो इसकी replication है, it is semi-conservative. It is semi-conservative. Now, what they have done, they cultured this. Yeah, they reproduce this E. coli, or they reproduce this E. coli in a cultural medium containing what we call as heavy nitrogen. They cultured this E. coli in a cultural medium containing heavy nitrogen, and this what we will see in this what we call as this, in this heavy medium जो nitrogen है, that is the heavy nitrogen, and this heavy nitrogen, it is provided by ammonium chloride. Again जो यहाँ पर नाइट्रोजन है, that it is heavy nitrogen. So what did they do? For many generations, this nitrogen, it was carried on this heavy nitrogen. Now, when this was done, it was found that this what we will see after replication, what they have found that both the strands of this DNA of this E. coli, it contains N15. That both the strands of, after many generations, for many generations, they have cultured this E. coli in the heavy this heavy nitrogen, and they have found that both strands of this DNA, it contains N50 as nitrogen 15 as constituent of किसके, जो प्यूरीन्स हमारे पास नाइट्रोजन बेस से, जो प्यूरीन्स and constituent of pyrimidines. Now, this heavy DNA, इसमें हम क्या देखते हैं कि जो नाइट्रोजन बेस है, they are N15. That is that this heavy DNA molecule, it can be distinguished from the normal DNA. This heavy DNA can be separated from normal DNA by centrifugation. by centrifugation in cesium chloride. By centrifugation in cesium, what we call as cesium chloride, CaCl, cesium chloride density gradient, जो हम इस्तेमाल कर लेते हैं, that is a density gradient. And what we will see, जो ये वाला है N15, it is not a radioactive isotope. It can be separated from this normal isotope simply by on the basis of the density. Now, what to understand this, what is happening, कि क्या हो रहा है, what they had done, let us see what is happening in this experiment. When the bacteria which were placed in the N15, what to understand this, what they have done, what is happening in this. For example, if this is the one strand, is the second strand, this also contain N15, this also contain N15. That is right. These are the DNA, two strands of the DNA. When they were cultured, now जो हमने medium, जो यहाँ पर हम क्या देखते थे कि these were cultured in the heavy nitrogen for many generation. Now we got this. Both strands, they were N15, heavy nitrogen. Now, when this heavy nitrogen DNA, they were cultured under optimum conditions, what were found, and what we will see, जो हमारे पास culture में वह हमने change कर लिया. We have taken this normal, and जो यहाँ पर हमारे पास जो रिजर्वायर है जहां से हमको ये नॉर्मल नाइट्रोजन आता है, दैट इज अमोनियम क्लोराइड, एंड दिस अमोनियम क्लोराइड इज आल्सो प्रोवाइडिंग अस द नाइट्रोजन, दैट इज द नॉर्मल नाइट्रोजन. एंड व्हाट वी फाउंड, जो रेप्लिकेशन हो जाती है, लाइक दिस इज N15, again this is N15. Now, what we will see, another one. इसका जो दूसरा strand है, that will be N14. इसका जो strand होगा, that will be N14. This is the new DNA synthesis जो हो गई इससे. Now, when they were again, अब हमने इनको फिर कल्चर किया in the optimum condition. Here again, जो हमारे पास जो नाइट्रोजन है, दैट इज द नॉर्मल नाइट्रोजन. सो व्हाट विल बी द रिजल्ट्स, से यहां से एक से हमने ये देखे, अब पहले हम ये वाला स्ट्रैंड देखेंगे. व्हाट विल बी द रिजल्ट्स, सो सी हियर, दिस इज द N15, जो सेकंड होगा दैट विल बी ऑफ N14. ये वाला है. नाउ टेक दिस वन, रेयर वन, N14, again this will be N14. Now take this one, this will be again N14. These are the new strands of DNA, जो इसका लाइफ स्पैन है 14 मिनट्स के बाद. अब हम ये वाला लेंगे, again this will be the N14, and इसका दूसरा स्ट्रैंड जो होगा, that will be the N15. So when we have done, हमने क्या किया, that one notes that it contains one strand heavy nitrogen and second strand जो हमने देखा नॉर्मल. यहां पर हमने देखा एक स्ट्रैंड हैवी है, दूसरा स्ट्रैंड दैट इज नॉर्मल. एंड when these hybrids, अगर हमने उनका क्या, when they were again what we will see, they were separated independently. Then they were again, उनकी जो generation हमने continue कर ली, and they were replicated under, they were cultured in this nitrogen normal जो हमारे पास है net N14. We will get these trends after 20 minutes जो उनका life span है, we get this. And again we will see that this gives both these trends. This one same normal, this वाला normal दोनों strand, and this one एक normal एक heavy isotope दिया गया. Now what we will see कि out of the 4 यहां पर हमारे पास DNA बन गए. It was observed that out of total 4 DNA molecules, 2 were completely, this one and this one, they were normal, or they were non-radioactive, and the remaining 2, this one and this one, last one, उनमें हम क्या देखते हैं, half radioactivity, half non-radioactive. एक heavy है, दूसरा normal है, एक heavy है, दूसरा normal है. Now what did he did? उसने क्या किया? And what did he did? The various samples जो हमने यहां पर देखा, they were separated independently on cesium chloride gradient, density gradient. वहां पर हमने क्या देखा? In order to measure the, क्या मेजर करना है? Density of the DNA after every 20 minutes. So what we will see, if this is the syringe, first one, like this, if this is the syringe, what we will see, this one, दोनों स्ट्रैंड जो हमारे, इट्स सीसियम क्लोराइड इसमें हमने डाला, सीसियम क्लोराइड. यहां पर हमने पहला वाला क्या देखा? N15 एंड N15. नाउ जब हमने दूसरा स्ट्रैंड ये ले लिया, ये वाला, इस कंडीशन में हमने क्या देखा कि इसमें भी हमने सीसियम क्लोराइड, हमने सेंट्रिफिकेशन कर ली, इसको शो करने के लिए, एंड वी गेट N14 and N15 strands. Now in the third जब हमने बनाया, थर्ड इस सीरीज में हमने क्या देखा? This is the third series. Here we will see again what we will see, in this strand, what we will see, sorry, दोनों यहां पर हमने क्या देखे? N14 and N14, and in the second part what we will see, N14 and N15 यहां पर आ गए. So what we will see, when this culture medium, when this E. coli DNA strands, they were cultured on the medium which contains heavy this nitrogen, and when they were cultured and we get them many generation, and both of them जो उनके DNA and they have got this heavy nitrogen. When this heavy nitrogen, वो वाली सेल जिनको हमने कल्चर किया under optimum condition, वहां पर अब हमने कल्चर चेंज किया, वी हैव पुट द नॉर्मल नाइट्रोजन, एंड दिस जो हमारे पास जो सोर्स है जहां पर हमको नाइट्रोजन, दैट इज अमोनियम क्लोराइड, वी गेट दिस वन स्ट्रैंड हैवी एंड अनदर स्ट्रैंड नॉर्मल है, वन स्ट्रैंड हैवी एंड वन. जब रेप्लिकेशन हो गई, 2 से बन गई 4, 4 से बन गए हमारे पास 1 से हमको बन गए 2। अब यहाँ पर 2 से कितने बन गए 4। जब हमने इनको फिर कल्चर कर लिया अंडर नॉर्मल कंडीशंस, इस वक्त हमारे पास क्या है, जो कल्चर जो हमारे पास है, दैट इज द नॉर्मल नाइट्रोजन। वी विल फाइंड हाफ ऑफ देम, वी गेट 4। 2 से हमके कितने बन गए 4 स्ट्रैंड्स में हमने क्या देखते हैं, 2 दे आर नॉर्मल नाइट्रोजन स्ट्रैंड्स, लेकिन एक में हम क्या देखते हैं, हाफ नॉर्मल हाफ हैवी, हाफ नॉर्मल हाफ हैवी। लेकिन so this means half is made up of the older and half is made up of the new. So this is called as a semi-conservative medium. Now the various samples, they were separated independently on cesium chloride gradient to measure the densities of the DNA after 20 minutes, during a second replication after 40 minutes, in normal and 14 conditions. What we will get, both the strands again separated. हमने यहाँ पर देखा they separated there, यहाँ पर दोनों हैवी है, यहाँ पर हमने क्या देखा, they separated again. So it was out of जो हमने देखा 4 strands जो हमारे पास आ गए, that out of the total 4 DNA molecules formed, 2 were completely these normal and 2 were what we called as the half heavy half normal. So this experiment हमको ये दिखा देता है कि that जो DNA replication है, it is semi-conservative. This was all about this experiment. Tomorrow we will study another experiment which will try us to show that the DNA replication is semi-conservative, and that was given by the Taylor. Until then, goodbye. God bless you. Thanks.